It's summer as I record this video, the perfect time to observe Fraunhofer's absorption lines in the solar spectrum. To make this observation, you need clear skies and a visible sun. The minimum required equipment includes a holographic diffraction grating film and a thin, adjustable metal rod, such as a radio antenna, to reflect the sun's rays. Here is a diagram of the experiment, along with the observation result captured using a Canon EOS 250D digital camera. The tube enhances contrast, but you can achieve a striking result by observing the virtual image against a dark, matte background in the shade. The result captured with a digital camera is somewhat disappointing. Color reproduction isn't very accurate, as the camera struggles to render monochromatic colors, like those of a rainbow. Two concepts explain this phenomenon: gamut and metamerism. The gamut refers to the range of colors a system can reproduce. Metamerism, on the other hand, refers to the phenomenon where two lights with different spectral compositions can produce the same color perception to the human eye. In simple terms, digital cameras are designed to reproduce polychromatic colors found in nature or daily life, mimicking how humans perceive colors. If you're curious about this topic, I recommend Steve Mould's video, Why You Can't Take a Good Picture of a Rainbow.
Oh my god, it's full on! Double rainbow all the way across the sky! Oh my god! Oh my god! Oh god!
Let's return to our main topic, spectrometry. We'll now take a look at how a laboratory spectrometer works. Here, I'm using a USB2000 spectrometer, which was marketed by Ocean Optics in the 2000s. The optical system includes a 100 micrometers rectangular slit, mirrors, a reflective diffraction grating, and a Sony ILX511 linear CCD sensor, optionally paired with a semi-cylindrical lens. Light enters the spectrometer through the slit and an optional filter. It is then collimated by a mirror, before being reflected by the diffraction grating. To make the light beams visible, I use an ultrasonic humidifier filled with water. The light source is a white LED. If you look closely at the image formed of the sensor's position, you'll notice a significant drop in light intensity between blue and green. This phenomenon is characteristic of phosphor-based white LEDs. The diode emits blue light, which is then partially absorbed by a phosphor layer that re-emits light in the green, yellow, and red wavelengths. This spectral mix produces white light, with a warmer or cooler tone depending on the phosphor formulation. This is another example of metamerism. Different spectral distributions can produce the same perception of white for the human eye. I'll now close the casing and show you some experimental measurements taken with this spectrometer. Here's the spectral distribution of the Sun. One can distinguish certain Fraunhofer lines, notably the one around 760 nanometers. This corresponds to the telluric oxygen absorption band. Note that the spectrometer is not calibrated. If you want to explore this subject further, I have made a measurement of the solar spectral irradiance using a calibrated spectrometer. You can check my graph on the English Wikipedia page dedicated to Fraunhofer lines. Let's now look at the spectral distribution of a phosphor-based white LED, the same source as before. Blue light is emitted by the diode around 440 nanometers. The phosphor re-emits over a broad spectral band around 550 nanometers. You can now understand why we observed a dark band between the blue and green regions, around 470 nanometers. To conclude this video, I'll perform a fluorescence experiment with extra virgin olive oil and a blue light source. Under ambient light, the olive oil has an amber yellow color. When illuminated with a blue LED emitting around 400 nanometers, it appears as a beautiful reddish-orange, close to coral red. I'm not sure if this color description is entirely accurate, so take a look at the result recorded with the spectrometer instead. The fluorescence peaks observed around 680 nanometers and 720 nanometers are well documented in the scientific literature on adulterated olive oils. The one I used here, and the one I cook with, is of high quality, I'm convinced. This isn't a scientific assessment, just a gourmet's appreciation. If you enjoyed this video, feel free to explore my other videos on my YouTube channel. I cover a variety of technological topics such as optics, silicon microsystems, and mechanical watch movements. You'll also find several useful links in the description covering spectrometry, radiometry, photometry, and color perception, including links to my notebooks published on Observable about the CIE chromaticity diagram.